to the inner membrane.
Chapter 4, Cell Structure and Function. The main student learning objectives for this lecture is for you to understand cell theory first. How did they discover that life is made up of cells? So we'll go over the discovery of the cell theory. We'll study the main structures in eukaryotic cells. That is you and me. Plants, animals, fungi, they are eukaryotic. So the main structures and the main functions in a eukaryotic cell. Next class, we'll study about cell membranes and the function of cell membranes. So how do we study cells? See, cells are microscopic. So if you look in this picture here, see, our eyes can see up to 100 microns. You know, if you have like 20/20 vision, you can see clearly. You can see a frog egg, 1 millimeters. But most of the cells we are going to be studying is about 50 microns, 50 to 100 microns. See the plant cell and the animal cell. So you need a microscope for it. Whereas bacteria and mitochondria, they're only 1 micron. So you would need a microscope. So you'll be using a compound light microscope for this class. But most of the pictures in your book are electron microscopy pictures. See here, if you see the structure of virus and stuff, they're only 100 nanometers. Then we'll use an electron microscope. So after the discovery of the microscope, you know, everything just took off in biology. Most of the discoveries were made after they discovered the microscope. It's such a fun instrument, you know, you can spend hours on it. So here's a picture of a compound light microscope. So they're called light microscope because we are using the properties of light, like refraction and refraction, to magnify the image. So this can magnify up to 1,000 times in our labs. There's also 2,000 in some labs. And this is called dissection microscope. Here, the image will be 3D. You can actually do the dissection, but very low magnification. And here is a picture of an electron microscope. So electron microscope are using the, instead of light, it will use a ray of electrons to magnify the image. So it can like magnify it like 1,000,000 times. So most of the pictures in your book are electron microscopy pictures. Cell theory. So what does cell theory state? All living things are made up of cells. Cells can only come from pre-existing cells, and cells are the fundamental units of life. And then, of course, it took years to discover this. The first person who coined the name cell is Robert Hooke, see 1635. He observed Koch cells under the microscope, and it looked like a cell or a roof with a person in it. So that's how it got the word cell. The study of cells is cytology. This is what he looked at. So here's Robert Hooke. And then Anton van Leeuwenhoek. His contribution is also, he made a lot of contribution. He's like father of microscopy. And then there's Schleiden, who proved that all plants are made up of cells. And then Schwann proves that all animals are made up of cells. And then it's Virchow and Remarque, who proved that cells come from pre-existing cells. It's not spontaneous generation. It just doesn't come from air. One cell can only come from another cell. So you can see it's a span of like 200 years for this theory to take root. It's a very, very important theory. Just like how atomic theory is super important, it shook up the whole world. This shook up the whole world too. So when we are studying cell structure and function, they are so diverse. If you just take the human body, right? The cells that make the eye are very different than the cells that make your heart or your kidneys or your spleen. So there's this huge diversity, but The basic organelle structure and function is the same in all of them. So macromolecules will come together to form organelles. These organelles will all come together to make the cells. Cells come in various sizes, shape, and function. But in general, right, you can take any cell, like a bone cell or a blood cell. All of them will have a plasma membrane. The matrix is called as the cytoplasm. It will have DNA as its genetic material and ribosomes for protein synthesis. There are two basic types of cells that evolved from the common ancestor. The first to evolve is called prokaryote. So this is where bacteria will fall. Pro means before, karyote means nucleus, before the evolution of a nucleus. So bacteria and archaea are prokaryotes. All the others, fungi, plants, animals, protists, they are all eukaryotes. And this is what we're going to focus on today, the eukaryotic cell structure and function. So before we go into it, you have to understand the difference between a prokaryotic cell like bacteria and a eukaryotic cell. The first difference is here the DNA in a prokaryotic cell it's called naked DNA. It's just hanging there. It's not protected in a membrane. That region is called nucleoid region. Size difference. See, it's a bacteria or a prokaryote, it's only 1 to 5 micrometers. Whereas a eukaryotic cell is much larger, more complicated, more sophisticated, 100 to 100 microns. The ribosomes, which are protein factories, their size will vary in both of them. In prokaryotes, it's called sedimentation coefficient. It is 70S. It's a little smaller, whereas it's 80S. Eukaryotic cells will have a lot of membrane-bound organelles, which is absent in the prokaryotic cell. So here's a bacteria. You can see how the bacteria is protected with like a capsule, a cell wall, cell membrane. The inner matrix is called cytoplasm. It has ribosomes hanging around for protein synthesis. But other than that, it doesn't have mitochondria and all that. It is simple, unicellular, means single cell. Always remember, it does not have a nuclear membrane. Our focus today is the eukaryotic cell. So here is a plant cell, and here is an animal cell. So you can see how they differ. So how are your cells in your body different from a plant cell? So you can see in a plant cell, it has a cell wall made of cellulose. It has this huge storage vacuole where it's going to store water and like proteins and stuff. And then what are these green structures called? Chloroplast. Chloroplast, excellent. Right. Sorry. So you can see, thank you, Stefan. So you can see chloroplast. So these are the three main differences between plant cells and animal cells. So we'll study those structures in detail. It's easy if you have the analogy of a factory. If you think of the cell as a factory, you know, like for example, the cell wall can be the walls of the factory, protecting the factory. The security guard is the cell membrane. And then the site of where all the activity is happening is the cytoplasm. So this analogy might be useful. You can come back to this. So first we'll approach a cell. We are going to zoom into a cell. So in an animal cell, the first thing you'll see is the plasma membrane. We have a lecture on plasma membrane very soon. Next lecture is plasma membrane. This is a Nobel Prize winning discovery model. It's called fluid mosaic model. The macromolecule that makes the membrane of our cells is called phospholipid. Because there are two layers of it, it's called phospholipid bilayer. The whole membrane is not static. It is dynamic. It's like moving back and forth. It's in a state of flux all the time. So that's why it is called fluid. And then it's studied with a lot of proteins. So it's called mosaic of proteins. Each protein will have a function. So we have an entire lecture on this. So now plasma membrane is the ultimate barrier of a cell. It is going to protect the cell. It is selectively permeable, that word. Selectively permeable, meaning it is going to regulate the flow of molecules inside the cell. See, the cell's objective is to maintain homeostasis. Only the nutrients that the cell need will come into the cell. So the phospholipid will have a phosphate group and two long fatty acid chains. And this is water loving. So this is hydrophilic. And this part is hydrophobic. And because of this nature, it forms a bilayer like this. Then you can see all these proteins. The protein that goes right through is called integral protein. The protein that sits on the outside is peripheral protein. They will attach to structures called cytoskeleton to give it strength to the cell. There's cholesterol molecules, which is very important to maintain the fluidity of the cell. And then you can also see some proteins that are carbohydrates that's attached to the protein. So it's called glycoprotein, which will play a huge role in antigen-antibody reactions. Glycolipids play a huge role in cell communication. How do cells communicate with each other? So this is a very important picture of the cell membrane. Then right next to the cell membrane, inside the cell, there's a jelly-like matrix that is called cytoplasm. So between the cell membrane and the nuclear envelope, there is the cytoplasm in which all the different organelles are suspended, usually on these filaments that you see here, like mitochondria, all of them. So what gives structure to the cytoskeleton? There are these proteins that form all the framework of the cell. They are called cytoskeleton. There are 3 types of cytoskeleton, and they are divided depending on its size. So you can see microfilaments. A good example is the actin filament seen inside our muscle cells. They are thinnest. They are also found in microvilli, which are like little projections in a cell. Intermediate filaments are intermediate in size. This is actin, 7 nanometers, intermediate filaments. And they are the ones who will anchor all the organelles in a cell. Microtubules are the thickest. They will play a role in cell division. So when we study mitosis, you're going to study this. They can polymerize, so they can monitor the movement of chromosomes during cell division. So cytoskeleton are proteins that gives structure to a cell. It will keep all the organelles in place, and it will allow organelles and vesicles to move inside a cell. So here's fluorescent microscopic pictures. You can see the 3 different cytoskeleton elements. Then we move on to the endomembrane system, all the membranous organelles that are found in a cell. So the main membranous organelles that you see in the cell would be the nucleus, the nuclear envelope, endoplasmic reticulum, that structure. It can either be rough or smooth, and Golgi apparatus, and lysosomes and vesicles. So these membranes will play a role in modifying and packaging proteins and transporting proteins. So if you take the nucleus, it is protected with a two double membrane. And then it will have these pores called nuclear pores. The main function of the nucleus is to protect the DNA. So you can see how DNA is condensed, and you can see DNA, which is the genetic hereditary material protected in the nucleus. The site of the matrix of the nucleus is called nucleoplasm. And the darkly staining structure is called nucleolus. This is where ribosomal RNA is made. So this is like the boss of the cell. It's going to control all the activities of the cell. The next endomembrane system is endoplasmic reticulum, ER for short. So you can see how in some it is dotted with ribosomes. So it is called rough ER. And then there's some ER without ribosome, it is called smooth ER. So in rough ER, so they will play a huge role in protein synthesis and phospholipids also. They make the phospholipids for cell membranes. So what happens is these ribosomes will make the proteins and they will be modified here a little bit. And then it will pinch off in a vesicle and it will go to this organelle called Golgi, where it will be processed. Smooth ER does not have ribosomes. They will play a role in the synthesis of carbohydrates, lipids, hormones, but it's also like the spa of a cell. It can detox poisons. Like people who drink a lot of alcohol will have a lot of smooth ER in the liver cells to get rid of the toxin that's in their blood. So now this protein, when it moves on, see, it pinches off in a vesicle. It moves to the Golgi apparatus, named after the scientist. The part of the Golgi that's facing the nucleus is called as the cis phase. The part that is away facing the cell membrane is the trans phase. So when this protein vesicle fuses with the Golgi apparatus, it will move through all these series of tubes. And as it goes through all these tubes, it will be modified. So that's why it is called as the shipping and handling center of the cell. So it will put a little sequence on the protein that will tell the protein where to go. So if it's, let's say it's a hormone, and then it will leave the cell. It will leave the cell, maybe out into the blood that's there. Or if it is a protein that has to go to the nucleus, it will put a sequence in it that will ask the protein to go to the nucleus. So these vesicles will take the new protein to the various organelles in the cell. Here's an electron microscopy picture of the apparatus. So main function of Golgi is protein processing and directing the proteins where to go. It's like putting a zip code on a lector, 01007, it will take it to Belchertown. That's what Golgi apparatus will do. Lysosomes are like the cleanup crew in a cell. They are the janitors. See, a student once told me, see, when she sees this word lysosome, she's reminded of Lysol, the cleaning solution. That's what lysosomes do. They clean up the cell. They'll make sure the cell is sparkling clean. All the dirt has to be gotten rid of. If there is an old mitochondria, that has to be recycled. So what does it have? So the lysosome is filled with harsh digestive enzymes that can break down proteins, sugars, lipids, nucleic acids, or even old organelles. So here's a picture of a bacterium that's invaded a white blood cell. So the bacteria has entered the cell. So what lysosomes will do is it will go fuse with it and digest that bacteria. So this is phagocytosis that's happening in our cell. So when you get a cut, a bacteria enters your bloodstream, all our white blood cells do this. This is the constant battle that's happening in our body and we never see it. Peroxisomes is another organelle that will get rid of the peroxide. Peroxide is a byproduct of metabolism. So peroxisomes will oxidize and break down hydrogen peroxide, and also it can break down fatty acids. And it can also detoxify poisons. So that's another organelle that's found in the cells. Ribosomes are the protein factories in a cell. So it will have a large part, they'll call it the large subunit, and it will have a small part called the small subunit. And the main function is protein synthesis. It will be able to read a template of RNA and then make a copy of proteins from that. Mitochondria, commonly referred to as a powerhouse in a cell. It has double membrane, a smooth outer membrane, and the inner membrane is like thrown into folds called cristae. And then there's an inner mitochondrial space. And then there's the mitochondrial matrix. The unique thing about mitochondria is that it has its own DNA, like circular DNA, and it can make copies of itself by binary fission. It can pinch off into two. So this is where cellular respiration happens. So what is cellular respiration? So here's the equation for it. Sugar molecule in the presence of oxygen is broken down to carbon dioxide, water, and energy, ATP energy. And energy is required for a cell to be alive. No ATP, no life. The next organelle is chloroplast. Again, it has this beautiful structure, double membranes. And then it has all these stack of membranes called thylakoid. So this is where chlorophyll is. The green, plants are green because of chlorophyll. So that is packed in the thylakoid. The liquid part is called stroma. And then it will be connected with bridges. So again, photosynthesis is a unique capacity of plants to make sugars from sunlight. So what plants can do is that chlorophyll is able to absorb solar energy or photons of light. It can absorb water from the dirt, nutrients in water, and then it also sucks up carbon dioxide from the atmosphere to make sugars. C6H12O6. So the two things that you and I need for survival, food and oxygen, is provided by photosynthesizing organelles. There's algae that can do photosynthesis. There's a blue-green bacteria that can do photosynthesis, and plants can do photosynthesis. So that's why regarding climate action and stuff, people want to plant trees in order to maintain the oxygen content in the atmosphere. And then when the plant makes all this food and it sucked up all this water, it will store in a structure called vacuole, very important. If this vacuole collapses, the plant will actually wait. Another thing is plants have a very strong cell wall made of cellulase, which is a polysaccharide. It will give strength to the plant cell. So what is found outside the cell? We call that the extracellular matrix. They have a very important function too. So the structures that you can see outside the cell, like all these sugars that are attached, sometimes it's collagen, apart from giving extra strength to the cell, they will play a huge role in cell-to-cell communication. And they also will play a role in keeping the cells together. See, similar cells will come together to form tissues. How are these cells held together? So they can be held together by any of these structures that you see here. See, plasma desmata is a channel between the cell walls of two plant cells. So through that channel, they'll be able to exchange nutrients. Tight junctions, desmosomes, these are actually proteins that keep the two cell membranes together. So for example, here's a tight junction. So it looks like somebody came and stapled the two cell membranes together. So it's actually a protein that will stick the two membranes together. Or it can have like a tongue and groove action. They are called desmosomes. So a projecting protein in one cell will fit into a groove of the other cell. Looks like somebody came and zipped it up. So that is called desmosome. So there's all these structures that can be found between cells for communication. There's something called gap junction. Anybody in health major, it's a very interesting phenomenon. Two cells, they'll be connected by a portal. It's called gap junction. You can see that in your heart cells, where the cells are highly connected. So when the heartbeat starts, the whole heart can beat as lup, dup, because they are so highly connected. They are in sync. So these connections are important for that. This is like one of my favorite theories in biology. It is called endosymbiotic theory. How did the eukaryotic cell evolve? So it is believed that chloroplast and mitochondria, because they have their own DNA, they can make copies of themselves, are believed to be prokaryotes. So the cell membrane invaginated, brought them in, and then by like symbiosis means win-win situation. Both the host and the organelle benefit by that association. It stayed, and that's how the modern eukaryotic cell evolved. So mitochondria and chloroplast used to be a prokaryotic cell, but they established a symbiotic relationship with the eukaryotic host. How was this confirmed? It's by sequencing the DNA, mitochondrial DNA and the chloroplast DNA, and they found it to be so similar to its bacterial counterparts. There's a very famous scientist, her name is Lynn Margulis, who actually like anchored this theory in biology. She's from UMass. So we'll study the next part next week, the cell membranes, in our next class.